to on. So we're going to record this session and share it out later on. So thank you everyone for joining us today for Twisted Pink's very first Lunch and Learn series. We're happy to have the representatives from Travera with us today. And my name is Caroline Johnson. I'm the founder and executive director of Twisted Pink. We're a nonprofit organization based in Louisville, Kentucky. And our mission is to provide hope and connection to people living with metastatic breast cancer. And we do that by funding bold breakthroughs in research and awareness of the disease. So we're very excited today to bring you some information about new technology that's available to metastatic breast cancer patients. And with that, I would like to invite you all to add questions into the chat during the presentation. We will go over all of the questions at the end, but we're going to let Cliff Reed present on the new technology that Travera has available to you as patients. And Cliff is the CEO of Travera. He attended MIT, Harvard, Stanford. In our chat before the meeting today indicated that he originally wanted to be in education and went into biotechnology after moving out to Silicon Valley. So we're very excited for his passion about the field and ability to bring some new technology to cancer patients. And then Dennis Watson is the VP of Business Development at Travera. And I had the pleasure of meeting Dennis in person a couple of months ago. And this has been a long process and talk about getting this together, but very excited to have you all here today and learn more about this new technology. So with that, I will turn it over to Cliff.
Great. Thank you, Caroline. We're delighted to be here today. So today I'm going to talk to you about our new technology for rapid cancer therapy guidance. And we're very pleased to be doing this in partnership with Twisted Pink. And you'll hear first about our kind of product technology, what we do, and then we'll talk about, if you're interested, how you can take an action to actually use this technology to benefit you. All right. So the key question we're trying to answer here is, how do we improve the treatment efficacies for advanced stage patients, right? The patients who really come to the end of the NCCN guidelines and don't have very good options available to you. So what I want to show is, this is the page of the NCCN guidelines for recurrent unresectable or stage 4 breast cancer. And I'll point out in this page, what you see is under first line, you see trastuzumab, that's Herceptin plus a couple of chemos. Second line, you see Herceptin plus a couple of HER2 inhibitors. And at those first and second line stages, it's pretty prescriptive about what to do. But when you get to third line, the situation changes pretty dramatically. And what you see here is 8 different drug combos of 12 different therapies. And then that box in yellow on the left that says, third line and beyond, optimal sequence is not known. So this is a very unfortunate situation for breast cancer patients. I will point out to you that all 61 cancers in the NCCN guidelines end with a page that looks exactly like this. So the cancer therapy guidance sort of ends for stage 4 patients. And that's the problem we're setting out to solve. How do we do effective cancer therapy guidance for stage 4 patients? Now, this problem was supposed to be solved by DNA sequencing. And I was actually the founding CEO of Complete Genomics, one of the DNA sequencing companies. And we thought we were going to crack the code of cancer and provide a really effective targeted therapy for every cancer. And it didn't work out that way. When it works, it's spectacular. It's Gleevec for CML, curative. Osimertinib for EGFR positive non-small cell lung cancer patients, 85% response rates, right? Unfortunately, those are the exceptions, not the rules. And really only about 10% of cancer patients get any benefit at all from the last 2 decades of developments of targeted therapies. So what we do in the absence of that kind of guidance based on genomics, we fall back to the NCCN guidelines. And the NCCN guidelines tend to work for about 40% of all cancer treatments. That's across all patients. In late stage patients, that number is closer to 20%, right? That's not personalized medicine. That's population medicine. And even the DNA sequencing is not really personalized medicine, because we're just grouping people by a biomarker, maybe something like BRCA1, BRCA2 mutations. And it's still a population of people. There's nothing terribly personalized about that. So what we want to do is personalize treatments to each individual patient, have an N of 1 treatment option. Ironically, the targeted therapies are now beginning to proliferate so much. So even if you have a DNA mutation that guides you to a therapy, there are a lot of therapies now for each mutation. And for example, in non-small cell lung cancer, there's 7 EGFR inhibitors. Which one do you use? Well, osimertinib's frontline, but maybe that's not the right one for you, and no one knows how to make that decision. So fundamentally, the problem here is there is a large and growing unmet need to match each and every cancer patient to the precise treatment that will work for them at their stage of cancer. Now, the other solution that's been proposed is clinical trials, but unfortunately, this actually helps even fewer patients than DNA sequencing. Only 5% of patients get into a trial. Most of them don't benefit from it. It ends up benefiting about 1% of all patients. This isn't what we're trying to do in personalized medicine. You're N of 1, not part of a large population, and when you deserve and we want to provide more individualized insight. Okay, so I'm going to describe about our core technology, and I promise not to drag you through too much physics and math. But what we're doing is kind of a new solution to an age-old problem. And the age-old problem is, how do we not use clinical trials and population studies to pick therapies for patients? How do we use direct measurement? How do we use the same method that works so amazingly well in infectious disease? If you have an infection and you go to your doctor, they don't turn to a thing that's like the NCCN guidelines and look up the results of a lot of clinical trials. What they do is they try it on you. They take the bacteria out of you, they grow them up in a dish, they hit them with a bunch of different antibiotics, they pick the one that works, and they give it to you. That works for about 99% of all infectious disease patients, not 20%, not 40%, 99%, right? We've been trying to do that in cancer for decades, and it's never worked. And the fundamental reason it's never worked is timing. What I mean by timing is, when you take live cancer cells out of a patient, unlike taking live bacterial cells out of a patient, they don't thrive in a laboratory. Live cancer cells, you pull them out of a patient, unless you take heroic actions, are dead in 2 days. Bacteria love to live in a lab. Cancer cells hate to live in a lab. And the problem with them living for only 2 days is that cancer drugs typically take about a week to kill cancer cells in a dish. So if you take live cancer cells out of a patient, hit them with cancer drugs, and they die, you don't know if they died from the drug or they just died from being in shock of being outside the patient. Now, companies have solved that problem, tried to solve that problem for many decades by keeping the cancer cells alive. Well, it turns out that approach has backfired completely, because the act of doing unnatural things to keep the cancer cells alive causes them to change. If you pump them full of cytokines and growth, you put them in this chemical soup that allows them to grow and keep growing. But by the time you're testing them a week later, they're no longer the cells that came out of the patient. So we have this fundamental timing problem, that the cells die quickly and the drugs work slowly. How do we solve that problem? Well, that's the problem we have solved. So MIT partnered with the Dana-Farber Cancer Institute about 10 years ago to figure out if there was a fast enough test so that we could just kind of measure the results of applying cancer drugs to cancer cells so quickly that the cancer cells don't have time to change. And that's what we've done. So now I'll tell you about the core technology. It's called the suspended microchannel resonator. It's a little semiconductor chip. You actually see a picture of it there on the end of a finger. And what that chip does is it weighs single cells with extraordinary precision, about 100 times more accurately than any other measurement tool in existence can weigh a single cell. And it does it not optically, not using a microscope, which is how every other weight measurement tool works. It does it using a very kind of fancy piece of physics around measuring resonant frequencies of oscillating cantilevers, which I won't take you through right now. But in terms of practical terms, we can actually measure the change in diameter of a single cell of about 5 nanometers. Remember back to your freshman physics? The wavelength of visible light, 400 to 700 nanometers. No microscope can ever see this change. It's that tiny. Okay? So with this wildly, powerful measurement tool, MIT and Dana-Farber said, maybe we can build the first effective test where we can test cancer drugs against cancer cells in the laboratory. And it was based on this. Everybody knows, well known, that dead cancer cells weigh a lot less than live cancer cells. In fact, they weigh about half of what live cancer cells, but they take a week to drop that half of their mass. And we know that cancer drugs, take a long time to kind of cause that loss of weight of the cell. But here's the trick: is if it's going to weigh half as much in a week, maybe it loses a little bit of its weight in the first day, while the cancer cells are still happy and healthy, right? They haven't died after they're going to be dead in 2 days. But if you can make these measurements in the first day, maybe that can predict whether or not the drug is going to work. And we've never been able to make this measurement before. It's just too small. That's the magic of this MIT invention, is finally we can make a tiny, tiny little weight change measurement. So we do that. And here's how it works. We take live cancer cells out of the patient. In our commercial usage, we actually run 20 different drugs by the cells. But my example here will just show you one drug. So if you take live cells out of the patient and split them in half, 5,000 cells in each of 2 halves, put them in a well of a plate, 2 different wells of a plate. Add drug to one well, don't add drug to the other well, and keep all the other conditions the same, and wait 24 hours. And during that 24-hour period, those cells are going to be changing, right? But at the end of the 24-hour period, what we do is we weigh each individual cell in each well. It's not a bulk weight measurement of the wells. It's weighing each individual cell in each well. That's the magic of our measurement technology. And what we get, and you kind of see a picture of it on the bottom right of the slide here, is you get these weight distributions. You get these bell-shaped curves of weight. It's because the cells are in all different stages of the cell cycle, so they all weigh differently. But you get a bell-shaped curve of the weights of the cells with drug, and a bell-shaped curve of the weights of the cells without drug. And you compare those 2 bell-shaped curves. And if those 2 bell-shaped curves are basically identical, I mean the 2 sets of cells changed the same over that 24-hour period, means the drug isn't working. Drug isn't doing anything, right? But if those 2 bell-shaped curves shift because the drug is affecting the weights of one of those groups of cells, that means that drug is beginning to kill those cells. It's going to kill them in a week, but inside of 24 hours, we can see this tiny little weight change that tells us that this drug is working. Okay, so that's a great laboratory experiment. It's beautiful. We run it, every day. The question is, does it predict patient outcomes, right? And so we have now run our first 49 patients actually through the system to see, hey, does this weight change measurement in a very short period of time predict patient outcomes? And the answer is yes. And what you see here is our first cells were multiple myeloma, the second set of cells were AML, and then the final set of cells are the solid tumors. And just because it's so much easier to run experiments over blood cancers and solid tumors. And across all these different types of cancers, what we see is we can typically predict at about 80% accuracy whether or not a drug is going to work for a patient. And that works in positive predictions and negative predictions, meaning positive predictions, hey, we think the drug is going to work. And in fact, the drug does work in a patient. And negative predictions, we think the drug isn't going to work and the drug doesn't work in a patient. So around 80% of accuracy on both of those. That is extraordinarily good compared to 40% in the NCCN guidelines for all cancer patients and about 20% in the NCCN guidelines for late-stage cancer patients. Now, this isn't a lot of data. It's only 50 patients. And what we're doing right now is running clinical studies, just beef up this data set and show that it works, broadly across lots of different cancers, and in particular in solid tumors, okay? You can see right now we've only run 17 solid tumors. That's changing rapidly. We'll run many more this year. And if you're willing, we hope we can participate and run your tumor through this system. It's a very broad-based test because what's the one commonality of cancer drugs? What do they do? I mean, they all have different mechanisms of action and different molecular pathways, but the one thing they all have to do is kill cancer cells. If they don't do that, they're not going to work. But that's what we're measuring. We don't care, what the DNA is like, what the proteins are like, what the metagenomics are like. We don't care about any of that stuff. We just say, if you put this drug on these cancer cells, does it work? And maybe we understand this mechanism and maybe we don't. But we're getting a true readout of whether or not these cancer cells from this particular patient at this particular moment in time are being affected by this cancer drug. So you can see a lot of different cancers, all the carcinomas, a lot of different cancer drugs. And we don't do everything yet. From the cancers, we don't do the lymphomas, the sarcomas, the glioblastomas. In the cancer drugs, we're not yet doing the antibodies. We're working on that. But what you see on the right side of the slide, kind of right middle, is we are doing the immunotherapies. So, you see Pembro, Nivo, Ativo, and Dostar there. It's a slightly different assay because in order to test the checkpoint inhibitors, you're not testing them against cancer cells. You're testing them against the human immune system. You're testing them against T cells. So what we actually do is take the T cells out of the sample when we can get them. You can't always get T cells, but when we can get the T cells, we take them out of the sample and we run the checkpoint inhibitors against the T cells to determine if we believe the patient is going to respond or not respond to the checkpoint inhibitors. This is what our lab looks like, just to give you an idea. So what we do is we take a live biopsy and primarily malignant fluids, pleural effusions of the chest cavity or ascites of the abdominal cavity, although we're doing tissue now as well. We'll talk about that in a minute. Take the live cancer cells out of the patient. We have this standard little shipping kit with FedEx pre-attached FedEx label and a tube inside the kit, or multiple tubes inside the kit, where the whoever is drawing the sample squirts the cells into our little tube, put it in the shipping kit, press a button that says turn on the chilling system, seal it up, take it to FedEx. Shipped overnight to us from via FedEx. The next morning, we take the cells out of the shipping kit. We plate them in wells. We add drug to some wells, no drug to the other wells. We wait 24 hours. We incubate those cells. Then we run them through our weight measurement system, measure the weights of all the cells, produce all these weight distributions, compare them all, write it out into a really nice report, and send it back to the oncologist. There are two real powers of this clinical study, this clinical test. I mean, the first is speed. You get a biopsy on Monday, your oncologist has the results of this in his hands on Wednesday. It's a 2-day turnaround test, 1 day to ship the cells, 1 day to incubate, and then measure the cells. And the second is size. We only need about 5,000 cancer cells for each drug that we test. So we could test a whole panel of 20 drugs with only 100,000 cancer cells. And this can be done, not only from a malignant fluid where we typically get many thousands of cancer cells, but also from needle biopsies, core needle biopsies, and potentially even fine needle aspirates, a number 22 needle, which is a very safe and non-invasive technique for collecting live cancer cells. Here's just a site chart, kind of a picture of the report. When the report goes back to the oncologist, it's really simple and straightforward. Page 1 of the report is, hey, we ran these 20 drugs for you. We scored them based on our measurement system, and we scored them between 0 and 100. 0 means we don't think this drug is going to do anything. 100 means, wow, we saw this pop response, a very strong response to this drug on these particular cancer cells. And then we also provide oncologists in particular who want to drill down and understand more about our test, and patients who want to drill down and understand more about our test, the score of each one of the drugs plotted against the actual physical mass change. You can report how much mass did these cells lose? And we do that in this little 2-dimensional plot. So that's what we do. A CLIA lab that takes live cancer cells, runs a 2-day test, and produces a nice report over 20 drugs about which drugs are actually affecting those cells and which drugs, for whatever reason, aren't affecting the cells. Now, what we've done is partnered with an organization called XCures to make this available to patients like you in the form of a clinical study. So XCures it's a CRO, and they're running these certified trials in a very unique way. They're not just running them at one hospital or 2 or 3 hospitals. They're actually doing an N of 1 platform. The patient can be anywhere, doesn't have to be at a specific hospital site. So they have their own study that's called X-Inform. And this kind of aggregates the patient's data, the electronic medical record data, into a very beautiful and standardized format. It allows doctors and patients to really see their patient journey in one place. It also enables XCures to use that patient journey and all of the information to go out and kind of search the internet, come up with what they call a care summary, and provide pointers to resources, which can include clinical trials matching. Now, Travera RTX is the study that we've added to XCures, and this enables you to have your cells managed or evaluated by Travera against many different cancer drugs under the XCures IRB approved clinical study. We will work with your doctor to make that all happen, but you can enroll in that study very easily. And once you've been through that study, the results are sent to your doctor, and then you can sit down and talk to your doctor about that. And I'll put the labels up a little bit later. So who qualifies? So first of all, you have to be planning to start a new therapy, because if that cancer therapy guidance isn't useful unless you're going to go on to new therapies, sometime in the next few months. We have to have access to your live cells, okay? So right now, in the XCure study, this is pleural effusions of ascites. It's malignant fluids in a body cavity. But Travera is now CLIA certified for tissue. It's not part of the XCure study, but it is CLIA certified, so we can run tissue samples. So if you don't have a malignant fluid and you're interested in this, reach out directly to Travera. Don't sign up for the XCure study. It's not ready for that. Reach out directly to Travera, and I'll put up that contact information in the last slide. The other thing is this assay must be ordered by a treating clinician. You can't order it yourself. The doctor has to order it. And it's currently available in almost everywhere. We're not yet CLIA certified for New York, Maryland, and Washington, D.C. And that's all in process, just kind of in paperwork. And that'll be done later this year. So here's the summary. This is free of charge. If you go on our website, it's not free, but if you do it through Twisted Pink, it's free of charge. You enroll in the X form in the Travera RTGX clinical trial. XCures, works with you to coordinate with your doctor about, the biopsy that we need and the results that are going to come back. We actually collect 3 vials of 50 milliliters each. So we want quite a bit of that fluid. But if any of you have fluid, typically when you get tapped, it's a half a liter or a liter. So 150 milliliters is very, very small. Tissue is coming, but it's not part of the study yet. You have to put that sample, those 3 tubes, back in our little shipping kit, press the button, and get it to FedEx that day. These cells die so fast. If you wait just one day to get them to Federal Express, we can't run the test. It's such a quick test and that's the power and the value of what we do is speed. So we'll run 20 therapies and the 4 immunotherapies if we get enough T cells out of the sample, and then deliver the results back to the physician. So next steps, you need to tell us if you're interested. You can, enroll directly in the trial yourself, and I'll put this up again. Or if you have tissue right now and you want to talk to Travera directly, you can reach out to Dennis. And here are the contact information for reaching out to Dennis directly. There's Dennis's email. And for enrolling directly in the study, there's the link to the study. So with that, a kind of rapid overview, I'm going to turn this back over to Caroline and answer any of your questions.
Okay, so I'll start going through some of the questions in the chat. I think I was the first one with a question. You had one slide up that showed the statistics of predictions. I think you said it was 80% for solid tumors. Do you have any trends on subtypes of solid tumors where your predictions are not in that 80%? So it would be the negative.
Yeah. We don't know yet. We just don't have enough data. We've only done 17 solid tumor samples. 8 of the 17 were non-small cell lung cancer. And then there were others were breast, lung, colon, panc, So we just have onesie twosies in these other cancers, which is why we're so enthusiastic about partnering with Twisted Pink, because you can provide us with samples that enable us to take the breast cancer set and expand it dramatically, and then be able to answer your question either, for a reason something's not breast cancer, or, yeah, this looks great. We're really able to do this. And we have to say, we don't know yet. Frankly, we're cautiously very optimistic because of the onesie twosies that we've tried in each cancer. They've all worked. And in our hands, a carcinoma is a carcinoma. It's an epithelial tissue cancer. We don't really care tissue of origin, but maybe there'll be something we'll learn in the process. So I don't quite know yet which cancers are going to be the best and which are not going to be so good.
The only one caveat I would add to that, Caroline, is that we've tested many more patients than the 17 that are in the data set. What we know is in the lab it works, right? We see those responses, we see differing responses with different agents, et cetera. What we don't have on those larger volume of cases is the outcomes data, which is really the important piece at the end of the day, right?
Mm-hmm.
Okay, and then my other question was, and I'll let some other people chime in if they want to. You're welcome to unmute and ask a question, but if you want to type it in the chat as well, we can go through there. How do you work with oncology professionals in this process? Do you speak to the doctors about your technology if a patient signs up, and what has their feedback been?
Yes, we talk to every oncologist and educate them about what we have and what we don't have, right? We're trying to be very straightforward. It's very early stage. It's very exciting and promising, but it's very early stage. But what we invariably do is we connect to the oncologist typically through the patient. And I will say, and you may run into some of this as you talk to your oncologist, this field has been tried over and over again. Literally decades of effort, some of them very well funded, some of them very high visibility, saying, wouldn't it be great if we had a direct measurement test of cancer drug effectiveness for each individual patient? And it's never worked. And it's left a very bad taste in the oncologist's mouth because it's been promised and delivery has failed so many times before. And as I mentioned, all of the previous efforts to do this, whether it was, Oncotech in the 90s and precision therapeutics in the 00s and Mitra Biosciences in the 10s and Notable Labs in the 10s. We know all of the failures. We're kind of students of this field. They've all failed for really the same reason. And the reason is they had to grow the cells. They had to keep the cells alive for a week or more in order to test them. And no one's ever come up with a method of growing cells without having the cells change. So rather than us trying to work on a method of growing cells and having them not change, we just said, let's make the measurements before the cells have time to change. That's a very different, fundamentally different technological approach. But the oncologists invariably say, oh, you're probably just going to do the same thing that everybody else failed at. So we always have an educational challenge with oncologists. Say, we're doing this really differently. And it's being done differently because we have a new measurement tool. And new measurement tools change science. What is DNA sequencing? That's a new measurement tool, right? It fundamentally changes science and fundamentally changes medicine. And we are so different than everybody else. We can, look people in the eye and say, we think ours is going to work when nobody else has had work because we're not doing it the same way. But often we have to kind of make that case to the oncologist because they've lived through all the failures of the past.
And I'm always happy to help with that process, Caroline. I've even had draft emails that other patients have used and have had a lot of success with it. They've said, hey, if this helps somebody else, feel free to share it with them. So, but I always ask to try to get connected to the oncologist up front whenever possible. And they've been surprisingly receptive. I will tell you, from 15 years working in genetics and genomic diagnostics, my expectation is that we were going to hit some real headwind here. But as I have conversations with oncologists about this test, they start exactly where Cliff just said. Thanks, but no thanks, Dennis. This has been tried. As soon as we get just a few minutes to describe how unique and different the technology is, and the fact that we're the first time any assay has ever measured chemo response against the patient's actual cells and not some later grown iteration of those cells, that's when they go, okay, you guys actually might have something here. This is worth pursuing. And it's been in general very, very positive.
And then, okay, so the Maggie asked a question: why not test while treatment is working to inform next treatment, knowing it will be needed at some point?
Yeah, we do that. So there's no pre-treatment exclusion here at all, because what we want to test is the real cells, the real cancer cells, whatever they're being bathed in at the time. And so we don't say, oh, there's a washout period of a week or any of the kinds of things that you'd see in a clinical study. We just say, send us the cells that you've got. We'll run a bunch of drugs against them, and we'll tell you how your cells respond to those drugs in whatever current state they're in.
And this is valuable too, because it can happen over time, right? We could do the test multiple times. Mm-hmm. You're on a new drug, you begin to progress on that drug, you're looking to switch to a different drug. The reality is cancer is heterogeneous, and different populations grow and die off in response to different treatments. And so something that you weren't shown to be responsive to 6 months ago or a year ago, you might be responsive to today. And so the test can be done more than one time.
Okay, and then we had a question from Terry. How can this be used if your scans start to show progression, but maybe you don't have a large enough tumor or fluid to run a test to get a sample?
Yeah, it's a real challenge, and we hit this all the time. And if there aren't, this is a live cell test. And if there aren't enough live cells, and that means about 100,000 cells to test a panel of 20 drugs, then we can't do it. Now, if you send us 20,000 cells, we'll run 4 drugs. And sometimes that's valuable too. But if we can't get live cells, there's really nothing we can do.
And then Shauna asked a question: has this? Oh, it looks like maybe a statement.
I was going to ask a follow-up on that.
Oh, sure. Go ahead.
Can you hear me?
Yes.
Yes.
Okay. Sorry, I'm using my kids' headphones because I'm having trouble with mine. Okay, so I'm on my 5th line of treatment, stage 4 breast cancer. It's currently working, no evidence of active disease. So I've got everything that everyone wishes for, right?
Uh-huh.
But I know the shoe is going to fall. It's just a matter of when.
Right.
So what I'm understanding from you is I would have to wait until a tumor is large enough that we have something to send to you. So I couldn't really use this as a proactive way to be like, well, I'm not going to trial 4 other medicines. What would be best right at this minute? Right now, the technology is not there, correct?
That's correct. Yeah,
Okay.
we need more cells than probably could be gathered from you through a biopsy. And specifically, one of the things we can't do is just take a peripheral blood draw, like, you know, something like the Garden 360 test or anything like that. There are just not enough cancer cells in peripheral blood for us to run our tests. So we really are talking about patients with active tumor, you know, either active solid tumor or active malignant fluids.
And I'll add as well, Shauna, that's where the turnaround time though can be really valuable. I mean, you know, this doesn't have to be a primary tumor site. It can be from a metastatic lesion. In fact, there's some argument to be made that it's actually almost better to you know, to test against a metastatic lesion, because that's really the stuff that you're interested in knocking down, you know, most directly. But you know, we've seen sufficient cell count come out of 20 gauge fine needle aspirations of a, you know, of a liver met. So it doesn't take a lot of tissue, but we do need those live cells, and if that starts to pop up, then yeah, you know, I mean, being able to get an FNA, or if there's a fluid situation, a drain, and having those results back within a couple of days to make that next treatment decision can be really useful.
Okay, and then that was Terry's question, but Shauna asked, how has this exciting technology been widely shared with the main oncology centers, for instance, Fred Hutch, MD Anderson, et cetera? And how are you sharing that out?
Yeah, well, we're doing a lot of clinical studies right now. So we have a clinical study in multiple myeloma, for example, running right now with Dana-Farber, Mass General, Mount Sinai, Weill Cornell, Emory, and City of Hope. We're doing a study with NCI, the Bethesda Cancer Surgery Center. We're doing a study with Boston VA. And so we haven't done MD Anderson, oddly enough. It's just kind of who we talk to in our relationships. So we're definitely sharing out our technology for all of those people. But what they want is a lot of clinical study data, and understandably so. And we're participating in all those studies. But what we came to the conclusion was there's clinical study method of collecting data and validating the test, and there's also real world evidence method of collecting data and validating the test. And so we want to offer both. We want to, you know, make this available to, for in particular, late stage cancer patients who are just out of options, recognizing the clinical validation set is pretty limited, but have us be able to say, hey, if you want to help us now, we want to help you now, and be able to run this test right now. So we're doing both. We're definitely working with kind of the major cancer centers, but we're also working directly with patients.
And we'll, you know, we're a very small team. You know, I mean, we're really just getting started. We just launched our CLIA lab officially early last year or middle of last year. So I'm the only real officially commercial guy in the organization. The rest of us are a bunch of PhDs from MIT and or Stanford in Cliff's case. And so I am absolutely reaching out to a lot of these different centers. As I noted earlier, I've been in the diagnostic space for about 15 years, so I do have a lot of relationships, and I'm reaching out to these people sort of one by one. But that's a slow process. And we started having patients that were finding their own information, that were finding us and coming to us. And so we use that as a trigger to say, you know what, if they're looking for us, let's go to some of the foundations and let them know how to find us. Because a lot of people like the idea of this test, but don't know that it exists or where to find it. And so that's really the intent of the kind of programs we're doing today with Caroline and Twisted Pink.
All right. And then Christine asked, I was on Keytruda and Abraxane in a study and had a complete response, but once I went off it, the cancer came back in a few weeks. Let's see, is there a way to measure how long you have been on a cancer drug to have longer lasting effects?
That's a great question. I'm afraid the answer is no. Our test really provides no information at all about kind of duration of response. All we're measuring is kind of response, no response. And so it's going to be up to the oncologist and the literature about how these drugs work to really kind of manage the duration of response. But as Dennis made an allusion to, finding, in a late stage cancer setting, finding a drug or two or three that generate a really positive response can be a great thing, almost independent of the duration. Because if you go on that drug and then you get the response that we predicted, and if it lasts, a month or two, or if it lasts, 8 or 10 months, when it stops responding, rerun the test. And probably what you'll find is that drug no longer does anything. Those cells are dead. But hopefully the next drug that's going after a different segment of that heterogeneous cancer, then it starts working. So you can just use this in sort of a cyclical method to just make this a chronic disease. I'm afraid we're unlikely to cure, but we potentially are very likely to just knock down the biggest cell population over and over again using a different drug each time that attacks a different part of that heterogeneous tumor.
And we see resensitivity, right? To the point that Cliff was just making, it's not uncommon that you find out that you're now showing sensitivity in the cells to a drug that you previously had tried maybe a year or 2 or 3 years ago and started to progress on. And the reality is that drug worked. It killed everything that it was targeting, but then in the time that you've been off of it, it's possible that some of those cell lines grew back up. And so it's not an uncommon thing. First you look at it and you go, well, I already failed on this. How are they saying I'm sensitive to it? This thing doesn't work. But the oncologists often look at it and go, hey, this makes perfect sense. We've probably just grown that cell line back up.
So that kind of goes along with the next question from Christy. And she says, I'm assuming that this test only confirms that treatment would be effective today. So if the cancer changes and the current treatment is no longer effective, then the test would need to be repeated.
Exactly right. Yeah, we're doing a very kind of a snapshot of what's likely to work today against whatever heterogeneous blends of cancers, subclonal populations are in the patient at this moment in time. So it has no sort of predictive value of how the cancers are going to change in the future. It's just a snapshot of what works today.
And you may have already answered this question, but Sally asked, has Travera worked with oncologists from Stanford Cancer Centers yet? What is the relationship like? Is it receptive?
I went to Stanford and I walked the halls over at the medical center a few times and discussed this in some length with a variety of the docs, Heather Wakely and Joel Neal in the non-small cell lung cancer page, and Stephanie Jeffries in breast cancer, some of you may know. And they're intrigued, but we've never found a project to do together. Partly they said, hey, if you want to do anything through us, expect the IRB approval to be more than a year. I'm like, we're not really interested in 1 year lead time, so I haven't. But we have wonderful relations with Stanford, but we have nothing specifically going on with them.
And this is one of the nice things about this particular trial offer through XCures, as Cliff outlined it. You guys really can go to this website that's listed here on the bottom and sign yourself up for this. Your local physician, your local facility doesn't have to be a participating site. It's a self-enrollment. Once you enroll, the clinical research team at XCures, who are really the ones managing the trial and the IRB and everything else, will reach out to your clinician and say, hey, your patient's enrolled in this trial. We need a tissue sample, or in this case, we need a malignant fluid sample. This is how that would go. And as Cliff also lined out earlier, right now, we are working with XCures to have tissue added into the trial protocol. Yeah. I don't know exactly what the timeline of that is going to be. So right now, to be able to enroll in that trial, you do have to have a malignant fluid, a pleural effusion, or an ascites sample that can be collected. But in the meantime, until we get that available, if you guys were interested and wanted to reach out to me directly outside of the trial, we're still happy to offer the test to you at no cost. And if it's a tissue-based test, and I'm happy to help coordinate you and your clinician to help make that happen if that's something that you're interested in. Our only real sort of ask in that is we're looking to collect real world evidence data if we can't collect it in the trial. And so we have a really simple online survey that just is about your experiences and your outcomes that we'll send to you at some point, after the test has been completed and treatment's been selected. It's about 10 questions. It takes 8 to 10 minutes to complete. It's very simple, but it just helps us get some feedback on what the experience and the process looked like.
Okay, it doesn't look like anyone else has any questions. If you do, just go ahead and unmute yourself and ask.
While we're waiting on that, Caroline, I will also add, I noticed somebody asked earlier about the slides. I know you're recording the presentation and that will be available to everyone. I'm happy to create a PDF version of the slide deck. A real PowerPoint version is like a massive file, but I'm happy to create a PDF version and send it to you. So anybody that's interested can get that from you.
Yeah.
But also anyone that's on the call can email me directly if you'd like a copy of it too. But I'll certainly send it on to you, Caroline.
Yeah, that'd be great. And I can send it out to whoever needs it or wants it. Just send me an email, those that are on the call, to info@twistedpink.org if you want the slides, and I'll send them to you.
Thank you for the information. It gives us hope. I'm curious if you all have had contact with Emory University in Atlanta.
We
Yeah,
have had
Okay.
contacts.
Yeah.
And again, that was our earlier work in the blood cancers. So we
Yes.
haven't done that in the solid tumors, but Emory is actually one of our 6 clinical sites for our multiple myeloma study that's ongoing now. So yes, we know Emory, and if there's someone at Emory, if your clinician is at Emory, let Dennis know, and we'd be delighted to reach out to them and do some education with them.
And I do have a personal relationship from past lives with Kevin Kalinske.
Oh, that's who I was going to say.
And Jane Mizell as well. So I have spoken with Kevin, with Dr. Kalinske about this briefly, and brought up the idea of maybe beginning a trial at Emory. They were just underwater from a trial perspective, so he asked me to reach back out in a few months. But I think if there was an individual scenario in which you were coming to him and saying, "Hey, I'd like to have this," I suspect he would be open to that, and I'm happy to work. But I've known Kevin for many years.
Oh, that's encouraging. Thank you.
Really nice guy too, by the way. You're with a great doc.
Thank you.
We did have one more question on whether or not you have a contact with Stephanie Spielman Breast Center at Ohio State Wexner Hospital.
Wexner, we did a call with Wexner, but Dennis, you want to answer that?
No, not really anybody directly there that I know well from my previous life. So, but there's a lot of people at Wexner that I have at least indirect relationships with. So again, anybody that's interested in reaching out to their physician or even just wants more information, if the trial doesn't make sense for you right now, even if you'd like to email me, I have a physician brochure that's a really simple 1-page, front and back thing that I can email to you to give you some basic guidance for that discussion. As Cliff outlined earlier, you tell your doc that this company tests drugs, against your cells in a lab, and the initial response is going to be, "This has been tried for 70 years. It just doesn't work." But keep in mind, this is a very different measurement tool, and that's what makes this unique and special. And so I always push for at least a conversation to describe the technology and the data to them.
Yep.
So, and I have one final question. How many people are you looking to enroll in your study, and like long term? Can you just kind of describe what phase you're at right now, and long term where you hope to be?
Sure. The XCure study is IRB approved for 200 patients across all different solid tumor types. So, and we've only enrolled about a dozen so far in that study. So we just launched that study late last year, and very early stage in the study. Myeloma study is 100 patients. The NCI study is 40 patients. We have Sequoia study out of here in San Francisco Bay Area. We've done 150, enrolled 150 patients. So we're in the hundreds, we're not in the thousands. But as Dennis said, we only have about 50 where we've gotten outcomes data back that we actually use to compute. Did we get it right or didn't we get it wrong? We're not saying did our lab measurements produce data? Sure, they all produce data, but did they predict the actual patient outcome? And that's where we've only gotten 50. So we're hoping this year to ramp that up to be much higher.
Okay, let's see. And then there was one that says, can you include the physician flyer with the PDF of the slide deck?
Dennis, that's
Absolutely. Yeah, it's I just responded to that in the chat, but
Oh, okay, you did. Yep.
it's a very large file because it's designed just from a data side, a data perspective, because it's designed for professional printing. So I might have to send it in 2 separate emails. It might not work with that and the PDF, but I will do my best. Either way, I'll get both files to you, Caroline.
Okay, that sounds great. Well, we're approaching 12:45. If we don't have any other questions, or if you do, you can feel free to send your questions to Dennis directly or to info@twistedpink.org, and we can get you an answer. Thank you all so much for being here today. There were a lot of comments about how patients are receiving hope from this, and thank you, Cliff and Dennis, for both being here today. Knowledge is definitely power, and it gives patients the ability to have some information to at least take into their physicians and ask questions about. So thank you so much. We hope to do this again.
You're very welcome. Caroline, thank you for the opportunity to present. We look forward to following up.
Absolutely. Thanks so much, everybody.
Right.
Have a great afternoon.
Thank you all. Bye-bye.
Thank you.